Hello and welcome to another lesson from Quick Science. This is IB Biology topic 1, and today we will be focusing on cells and sizes. This is following on from the resolution and magnification as well as cell theory. So what we're talking about with cells and sizes is that cells in themselves are relatively large to start off with, and then as we go smaller we have their organelles, then there's bacteria. Smaller than bacteria are viruses, then we have membranes such as a nuclear membrane, and then smallest of all we have molecules. So for example, we have here a bacterium, could be measured at 2 millimeters, and we need to be able to convert it into micrometers or even nanometers. This again is a development from the resolution and magnification session that we went through previously. So in order to calculate a cell's size, you need to view the cell through a microscope, and you need to know the diameter of the field of vision. So for most cases within a laboratory, the field of vision can very simply be measured by using a ruler. Very common mistake that students make is that this can be overlooked and requires something more complicated. However, you can always use a ruler. The thing with using the ruler is that you need to make sure the scale is correct. So one way in order to calculate magnification is you can do the size of the image divided by the actual size of the specimen. That will give you the magnification. On most drawings you will find that there is a scale to help you with the calculation. So in this case we have a 10 millimeters, and we need to convert that from millimeters into nanometers. Nanometers can often be seen written like this. So we take the magnification, which will equal 1000, and we will then divide that by 25 nanometers. 1000 divided by 25 is 40, so the magnification is 40 times. Please note as well that there is no units for magnification. There are no units for magnification. So really important thing to remember is the change or converting of units. So 1000 millimeters is 1 meter. If you had 1 millimeter, that is the equivalent of 1000 micrometers. Excuse me. And 1 micrometer is the equivalent of 1000 nanometers. So one of the things as well, once we've calculated the cell size using the magnification, the image and the actual size that we have seen using a ruler, we can then also apply it to the surface area to volume ratio. It's a really significant factor, and is one that can help with all sorts of homeostatic mechanisms within living organisms, which we would look at in topic 2. So the rate at which heat and waste production and the rate of resource consumption are functions of its volume. So if you think that someone may consume a large amount of food, or as humans we have a core body temperature of 37 degrees, most of that will be lost as heat. Reason for our core body temperature being at 37 is due to enzyme reactions and their optimal temperature conditions being at 37 degrees. We also have cell membranes. Every single cell in our body has a membrane, and cell membranes control effectively what goes in and out. This can be done through active transport, through osmosis, and also through diffusion. We will look at those in more detail in the remainder of this topic. So as the width of an object, such as a cell, increases, the surface area also increases. So the bigger the cell, the bigger the area it takes up. However, the volume will also increase, so the surface area to volume ratio decreases, and thus the rate will decrease as well. So here's some examples for you. As the width increases, the surface area increases, and that has a much slower rate than what we would desire. So for example, if we had a cell radius of 0.25 and a measurement of 0.5, that gives us a surface area to volume ratio of 2.4 to 1. At this point, if you wish to pause the video to double check these calculations, simply working out a ratio, you can just do the surface area divided by the volume and you should get 29 to 1, 0.4 to 1 and 40 to 1. So with the surface area to volume ratio, it needs to be taken into account the different cells that we have in our bodies and the different functions that they have as well. So large animals have more cells, not larger cells. Again, a common misconception by students that the larger the animal, the larger the cell. This is incorrect. Some larger cells have modifications that make them more efficient, such as the shape and infoldings. For example, if you looked at the skeletal muscle, it is known as a striated or smooth muscle. There are also nerves. We have motor neurons that again have different shapes due to their different functions. That's all for this topic. Please do like, comment and subscribe. If you have any questions, please do feel free to leave them in the comments section below, and I will do my best to get back to you as soon as possible. Thank you very much. Bye bye.